Hey everyone, I want to say thanks for attending our webinar today. This is a webinar that's in one of a continuing series. And today we're going to talk about reconstructing whole mouse brain volumes from serial sections for registration to the Allen Atlas and its coordinate system. So with that, I really want to extend thanks and give a warm welcome to renowned neuroscientist, Dr. Chip Gerfen, and MBF Biosciences' own computational neuroscientist, Dr. Brian Eastwood. So they're both on the line with us. And today, I just want to talk about, I'll introduce the workflow in a bit. But I do want to remind people we're going to be using the BrainMaker workflow in our NeuroInfo software during this. So that's going to be running, that's the software that you're going to see throughout. The couple of notes, there is a survey at the end. If you can take a minute to fill that out, that would be really fantastic, because that'll let us know what worked as well as what you want to hear in the future. I think one of our next webinars on this particular workflow is going to be on the cell detection part of thing, which is enhanced by deep learning or machine learning algorithms. So we'll do a deep dive on that. But so yeah, that's one thing. The other thing is we will try to get to all questions, time permitting. But if we don't, I'll have a link at the end to our forums where you can correspond with us as well as other people at MBF and even other users. And it's a very simple link. It's just forums.mbfbioscience.com. So I think with that, I just want to show an overview of the workflow again that we've been talking about in each one of these webinars. And this workflow is nice in that it uses something that's readily available to most neuroscience labs, and that's serial sections, as opposed to having a complicated clearing method with a really expensive imaging device on the back end of the imaging process. So this uses something that's available to most neuroscience labs, serial sections. So today, what we're going to go through is the first part of that, really getting my sections cut onto slides, getting them imaged into whole slide images, and then getting them into volumes that are appropriate then for doing things like cell detection and registration to the Allen space. So in the common, you'll hear us say CCF, or Common Coordinate Framework. And that's really the coordinate system, Allen Atlas. And so this will be the first part of that, really getting images that are appropriate. And Chip's going to kind of dig in on some advice and some of his. He does this all the time and has for many years. So he's going to impart some of his wisdom on this process today. And Brian and I will be jumping in at different spots with questions. But Chip, at this point, I'm going to give you the keyboard and mouse.
Okay.
And I'm going to go to the next screen, and you can start. So everyone, Chip Gerfen.
Hi, my name is Chip Gerfen. Anyway, as Nate said, I've been doing this for a long time, and I just want to describe today both the histology processing, the imaging, and then how we use NeuroInfo to reconstruct from serial sections whole brain volumes. An advantage of the technique that we use is that this is all done on free-floating brain sections, and which enables doing immunohistochemistry and things like that. And I just want to describe, how we go about this. Most are very important for doing whole brain reconstructions is to have your sections from the brain put in the serial order from front to back. And we use coronal sections. And so the way that we do this is we cut with a freezing microtome or with a microtome. We cut the sections. In the first section we have a tray that has 10 wells in it. We put the first section in the first well, second section in the third, fourth, fifth, sixth, all through 10. Then the 11th goes into the first well, 12, 13, all the way through. So what this does is it puts in each of these wells a 1 in 10 series. So these wells that we use, and I'll show the trays that we use next. Each of these wells contains about 1 ml of solution. So we can then mount these sections. It's trivial to mount the sections in order because it being a 1 in 10 series. So in the first well, this is the frontal cortex a little bit further back into the striatum, further in the striatum, starting into the thalamus, all the way through to the midbrain, and even if we went further to the medulla. Anyway, so it's trivial to mount these sections that are 1 in 10 series in their specific order. And so one way that we used to do this is we would mount the sections after they've been processed for immunohistochemistry, so you can see the stains and all of that, on separate sections on 10 slides, each slide containing a 1 in 10 series. And then these could be imaged and processed for whole brain reconstructions. However, what we do is that we take each of these wells, and instead of mounting them on the slides, we actually then mount them in serial order from the beginning before we go through the imaging. So in this case, what we would do is we would take the first well, the first section from the first well, and put that that on the slide. The second all the way through. So we would end up with the serial order of the sections from front to back would be put onto the slides directly. And what I'm going to be describing is how we use that to reconstruct whole brain volumes. Next slide. So these are the wells that we use to do the immunohistochemistry. And so these are wells that we actually, originally they were made out of plexiglass, and now we use 3D printing. And how to use 3D printing is going to be put onto the MBF website. But you can see that each of these, this is one that has 10 wells. And so each of these wells is 1 mil, and they've got little holes in the bottom, and then they fit snugly into a tray. And so this enables us being able to use basically 1 mil per well, or 10 mils for the whole tray. We can process a whole brain. Usually it's about 120 sections for a whole mouse brain in these wells, and do the serial ordering. And I'll talk a little bit about this. And so this is one we oftentimes actually do 2 brains at once. And so this enables us to put 2 brains into the same well. And this is 20 mils of volume. Antibodies are expensive, and so we minimize the amount of the volume that's required for the antibodies with that. Okay, so now I'm going to describe how we take those sections that have been processed and reconstruct whole brain volumes using the NeuroInfo software. So Nate, can you switch me over?
I'm going to make you Chip, the presenter now. And I will say that at the end, I'll put up a link that has a link to the 3D printing files that if you wanted to print these yourself, you'd be able to do that. So let me make Chip the presenter here.
So this is a 3D volume that's been reconstructed from serial sections. And so in NeuroInfo, there's the ability to do a 3D, to view in 3D. And the first thing is we can see in the coronal view, a top-down view, and a side view. I'm going to show or demonstrate using a brain that came from Nuo Lee and Elisa Thomas at in Houston, from Baylor College of Medicine. And this is a brain in which they injected AAV antegrade tracers into 2 regions of the ALM frontal cortex, a red tracer and a green tracer. And then this shows the tracing, the projections from each of these injection sites through the brain, and then in 3D. So in the coronal section, this is the coronal view, and it's showing, it's all the way through. And then this is the 3D view. And so you can see how we've reconstructed these sections that have been put in serial order into a 3D view that can be viewed. And so you can see the projection tracks from the frontal cortical areas down through the striatum. And even in 3D, we can view that, I can see that this is the contralateral striatum, where there's both the projection from the lateral and medial injections. This is the ipsilateral striatum, the basolateral amygdala. And in other views, just flipping this all around, we can see this is the superior colliculus, the projections into the thalamus, projections down into the pontine regions, and even down into the brainstem. So this is the 3D visualization of the whole brain reconstruction. Now that 3D view came from the series of coronal sections that were put in order. And so this is the view of the sections in the coronal view. And these are the same sections that were used to construct that 3D view. And so here we can see in coronal sections, the two injection sites, one in the medial ALM and the other into the lateral ALM. These are prefrontal or frontal motor cortical areas. And then as we scroll through the sections, you can follow the projections as they go through the striatum. So I'm crossing over to the opposite side. You can enhance in this view. Coming down into the further in caudal in the striatum, and then the projections into the thalamus, basolateral amygdala, parafascicular nucleus, subthalamic nucleus, and into the superior colliculus.
So Chip, these images,
these sections that we cut are 50 microns, and these are imaged with 10x objective.
Oh, so it's resolution that's sufficient for doing things like cell detection and characterization of these projections.
Right, and seeing the axons, and I'll show next. So anyway, so this enables us to scroll back and forth through the different sections to go to the area that you want, and you can see that they're well lined up one to the other. And then what we showed in the last webinar was how to take this whole volume reconstruction and register that to the Allen space. What today I want to talk about is how we use the BrainMaker function to actually make this 3D volume. So this is a single slide that of serial sections, and I took just slide number 4 from this particular brain through the thalamic areas. You can see the basolateral amygdala, the projections into the thalamus and all. And just to show that this was the first section on the slide, second section, third section, all the way through. A couple of things. One is that we can see that sometimes the sections are flipped one side to the other. One of the great things about free-floating sections is that they give really incredible immunohistochemical labeling. Downside is that you have to mount them with onto slides, and sometimes it's hard to tell the right from the left. A trick would be that we could put a slash in one side of the cortex. We don't always do that. But this software will enable us to flip these sections to actually orient them correctly. Another thing I want to point out is that we image these. I'm going to zoom into a small area in the thalamus. And we image through multiple focal planes. And now I'm going through the multiple focal planes. So we start at the top of the section. We image, like 5 microns down, take another image, and we end up with a stack of images through the whole section. Now, the advantage of that is that, at any given, at the top of the section, certain of these processes are in fine focus. But as you go through, they go out of focus and other ones come in focus. And so what we do is we collapse the stack of these stack of images for the whole slide using a process called deep focus. And Brian, who wrote the algorithms and software for this, is going to describe that now while I turn it on and run it.
You're turning it on. Okay, great. Yeah. So as Chip mentioned, this works on stacks of images, so focal stacks of images, where each plane is taken at a different focal position with the microscope. And what we do is we project from the 3D image stack into a 2D plane, is very much like a maximum intensity projection, except what we're using instead of an intensity for figuring out what to preserve in that 2D projection, we're using focus. So we evaluate focus everywhere in that stack. At each location, we select the plane where the focus score is the maximum, and that determines what information gets preserved in that 2D projection.
I think it's run its course.
Yeah, so it looks like you can just toggle it on and off, and you can change the focus when you're looking at the 3D focal stack, and then when you enable deep focus, you only have a single plane to look at, so the focus won't change.
So you can see in this particular plane, there's quite a bit that's out of focus, but then the deep focus is brought into focus the processes that were in other sections. So we can focus that on and off.
Yep, and that actually has done focus for the entire slide as well.
So this does it for the entire slide and all that.
What are you using to do that? Can you talk a little bit about the rig and tech you're using to do that scanning?
So we use a Zeiss wide field fluorescent microscopy, and we use actually other MBF software called NeuroLucida, which is we image with. And that imaging enables us to image. Let me zoom out a little bit. So that enables us to image a whole slide like this, and it images it, as I said, with a 10x objective. So it'll image as, a tile here, move over to an adjacent tile, and on each tile we image with depending on, and this is the case, it has 3 fluorophores, blue for the Nissl stain, green and red, but we've done up to 6 or 7 fluorophores. It will image each of those fluorophores at each plane and go through the whole, and then through the whole stack. Then it moves on to the next tile and does the same thing. So to image a slide like this takes about, it can take as long as 3 hours depending on the strength of the fluorescent signal that we're doing.
Cool, thanks, Chip.
Okay, so now what I want to do is to show that after we've gone through this and done deep focus and run the deep focus program on all of these slides, then we end up with all 8 slides now have been deep focused. And so this is showing the 8 slides for this particular brain. This is the first slide, second slide, third slide, fourth slide, fifth slide, 6, 7, 8. So the next part of this, the program, what it does is we're going to use BrainMaker, and we're going to take all of these sections. Now these are sections that have been put in serial order from the beginning. So these are all already in order. So this is, and they're ordered in columns. So this is the 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, all the way through to about 120 at the end of the brain. So the next step, and where we actually get to the BrainMaker workflow, which is here under BrainMaker workflow, is where we're going to take each of these slides, and we're going to then obtain each of the sections from those slides and then align them to each other. So for this one, I'm going to close all the files. So for the purposes of this, that the alignment process, doing the whole thing would takes about half an hour to 45 minutes. And what I'm going to do for the purposes of demonstration is I'm just going to take these 2 slides, the 4th and 5th slides, and I'm going to demonstrate how the program works on those. And just imagine that we've done that for the whole brain, for all the slides. So we start with the BrainMaker workflow. The first thing we do is add slides. That's the first step. So the workflows in the MBF software often has, outlines the specific steps for each process. And in this case, the first thing we do is we add slides. And I'm going to go to desktop. And this has all 8 slides. Actually, there's a couple of extras. It has all 8 slides, and normally I would pick all 8 of them and put them into the program, but for the purposes of demonstration, I'm just going to pick the 4th and 5th slide. Open them. So this is the 4th slide, and this is the 5th slide. And so normally I would have, slides from the front to back, but in this case, I only have these 2. Then we move on to, that was the first step. The second step is to select and order the sections. So I move on to this next step. Now there's 2 different options for how the sections could have been put on the slides. One is the staggered version. That's when I showed you initially that you could mount a 1 in 10 series, each 1 in 10 series on a separate slide, and then use a staggered format, and then that would take the first section and it would actually put them back in the appropriate order. However, for this one, we're going to use the sequential because that's how we've ordered them. There's a variable number of sections on each slide, and so that's what we select. There's a number of options to go through this, and if you ever use the program and have any difficulty with it, you can just call the NBF help people, and they'll be able to help you on that. So now we're going to view those 2 slides that have been put in here, and so normally, I've already done this for these sections in preparation for this demonstration, and normally I would there wouldn't be outlined, but let me just start here.
You can just hit apply and it'll recompute the outlines for you.
Okay, Brian, who wrote the software, always has to remind me how to use this. Anyway, so what it's done now is that it's outlined each of the sections. And so Brian's going to describe how this outlining works.
Yeah, so the goal is to as Chip said, define the outlines of each section. And when you're looking at whole slide imaging, usually each section is pretty well separated from the background. It's fairly easy to find how to separate them out. But here what we're doing is we're looking, in particular, we're paying attention to the boundaries, the edges of each section. And there are 2 controls that you can use to change how those contours are generated. The first is smoothing, so that's the parameter that you're looking at right now. And what smoothing does is it changes how tightly the boundaries adhere to the tissue sections, to the edges of the sections. And so larger values create smoother contours that tend to be pushed out further from the sections, and smaller values tend to create contours that tightly hug the boundary of the tissue. And I personally like to use a value somewhere around 2 or 2.5, somewhere in there. That tends to work well for the scale of the images that Chip is working with. Because what it does is it pushes the boundary just a little bit outside the edge of the section, so that just to make sure we don't lose any information, all of the all of the tissue gets included in each of those sections. We don't lose any tissue. The second parameter is a threshold. What that does is it controls how much of the slide is considered to be the foreground or the tissue. And that's, so if smoothness adjusts the local properties of the segmentation, then the threshold adjusts the global properties. And so if you're missing sections, if not enough sections are being found in your slide, or you're missing parts of sections, then lowering the threshold will include more of the slide. And if you're getting too much, the background is being included in the segmentation, then raising that threshold will work. So that is a property that affects the global segmentation.
So another thing now, in this example, the sections are all pretty well separated one from the other, but sometimes the sections are close to each other, and like in this case, and so the outlines are combined, and so these have to be split. So what we do here is to, oops.
You have to close that dialog first, hit OK, and then you should be able to select.
So now I've selected the boundaries for these, and you see that they're combined. And then I can right click and it will split those two. Okay. So then after these have been, let me go back and redetect.
You might want to change that smoothing back to around 2.5, somewhere in there.
Yep. So now we go through this process and it's detected and outlined the sections. And then what we're going to do is that the next thing is I'm going to, for the purposes again of demonstration, I'm only going to, I'm going to delete some of these that have been detected. Going to remove these, delete the selected content. So now there's 7 sections from this slide that have been detected. One thing we might do sometimes is that when there's like a little blib there, we can remove that.
I think that one, if you split that contour, I bet it'll find that little segment.
Yeah, it should. And you just delete it.
Yeah. So there's some cleaning up that can be done. Anyway, so now I've selected 7 of the sections from this, and we say we're done editing. And so now we're going to have it order. It's going to order the sections within the slide, starting with the ones that have been selected. So this is selecting the first section. Normally it would have selected this as the first one, but it's selecting this one, and then they're in columns. And so the second section, third section, fourth section, fifth section, sixth section, seventh section. And so this is the first on that slide, the 7th on that slide. So then we're done, and we'll go on to the next slide. And I've already done this. We had done the same process. And so now this first section in this slide is the 8th section in for the whole volume, the 9th, 10th, 11th, 12th, 13th. Again, this is the first section selected on that slide, second on that slide. So you can see how this does this. So it'll take the whole series of sections from various slides, and now it's put them into an order that's for the whole brain. And we've selected all of those. We'll go on to, and the order, how it does the ordering is by row or by column or manually. So we use the by column version. So now we've done the 2 slides for the example. We're going to go into the next step. So in this simple example, this puts all of the slides, all of the sections that have been ordered and in their order. So this is the first from that series through 13, and that sometimes you might make mistakes might be made, and so you have to adjust the orders of that. And again, this is something that's available. It requires a little bit more description of how to do that. Again, the excellent people at MVF can help with that. Those are all in order. So now that the sections have been selected, outlined, and put in order, we go on to the next step. And so this step aligns those sections, starting at the beginning and going all the way through the series. I'm going to start that using the top version. And Brian, who again has written this software of how to do this, is going to describe what's going on.
Sure. Yeah. So what's happening right now is we're using a procedure called image registration to align each section in the series to the preceding section in the series. And there are 3 options that are available here for how to do the alignment. They range, they basically do a trade-off between accuracy in the result and speed and how fast it operates. The one that Chip selected is using image features to do the alignment. And so what this does is it uses all of the available image information to align each section. And it works really well when the intersection spacing is reasonably small in between the sections. Then the result of that is that the neighboring sections appear fairly similar, even though the appearance of those sections changes gradually as you move through the brain.
And Brian, I'd throw out there too, like a general rule of thumb on that I found from working with people in their data is an upper bound on that of about 250 microns or so.
Yep. Yeah, that's right. Yeah, there needs to be some amount of the structure of the brain that's preserved in neighboring sections.
Between the sections.
Yeah. And what we're evaluating is translation and rotation to get those components to align. And then also we're looking at the flipping as well. So you remember that there's the injection site and the traced projections going through the brain. That sort of thing can be used, that's a big cue to being able to figure out whether there's flipping in this particular case. The second option is to align using just the shape. And what that does is it uses the outlines of the contours that we found during the segmentation step and just pays attention to that. It doesn't look at the internal content of the image. It's faster than using the complete, all of the image features. And again, it evaluates the rotation and translation to try to align those contours. And it also evaluates flipping, which can work if there's a little asymmetry, or for example, as Chip mentioned, if a notch was cut in the brain or something like that. And so even though there's not a ton of image information there, we can still evaluate the flipping and get it right most of the time. And then the third option is the fastest option, which is to align the centers of the section. It's the fastest because basically we find the center of mass of each section, and we just subtract the positions of those, and that gives us the translation between those sections. We don't do rotation, we don't do flipping in that case. But this is a really good option if the intersection spacing is very large, or there's a lot of tissue damage. And in that case, it can be good to just go to the next step where we do interactive alignment. You can initialize the alignment of each section interactively, and then run some automatic registration to improve the alignment. And we'll be moving to that step very shortly. It looks like we're just about finished up with the automatic registration here.
So again, depending on which of these three options one uses, it changes the amount of time. For in this brain, if there were 120 sections, which there were, it would take, it might take up to 15 to 20 minutes, or even up to a half an hour to do that. The most accurate, and doing it fast, it would take like 5 minutes. Anyway, so after we've done that, go on to the next step. Says the alignment was successful, click the next step. So this is where we review how well the automatic alignment is done from section to section. And this is going to show, as Brian said, that the way that this works is that each section is aligned to the prior section. This is the first section that's been aligned, so it's been aligned to itself. And there's a grid function here, which you might want to use to see, to just make sure that the section is aligned to the perpendicular grid. Usually turn that off, but that can be used to do the original one. So this has now gone on to the second section. It's been aligned. We can move back and forth between the first and the second. And now we can manually interact with those alignments.
So this is an overlay, Chip?
What?
This is an overlay of two sections, right?
Yeah, so this is an overlay of the second section onto the first, and we can see the transparency. So this is the second section. This is the first section. I use things like looking at the blood vessels, the shape of the hippocampus, ventricles are good to do alignment. rotating. So when I'm satisfied that that's well aligned, I move on to the next one. And the automatic alignment process has done such a good job that you almost can't even tell that they're not aligned. Now I'm rotating just to emphasize that there are in fact two sections there.
The other thing I just wanted to call attention to it is when you do make those changes, you can make small adjustments, or if there was some misalignment for a particular section, you can initialize the alignment. You can kind of get close and then hit the refine button. And what that refine button does is it runs a number of iterations of that image registration process that we just went through using the image content. So even if you didn't use the image content initially, you can use that at this point where we'll try to refine that, the rotation and translation, and just try to optimize it basically to get a better match.
Right. So I've gone to the next section. These are you can see that the automatic alignment process has done a very good job of aligning these sections one to the other. Just look at the way that the blood vessels along the midline line up and the ventricles line up.
The other thing to note is that the projections are all on the same side of the brain. In this case, it's the animal's right side, even though it's on the left side of the screen. And if you remember when we saw the whole slide images, you could see that those projections were on either side of the tissue as mounted on the slide.
So this is, I just unflip them. So this is the prior slide, and we're on section 7 here. And then say the next one was flipped, then we can flip it. Now, using the alignment process that's the most accurate, this is often taken care of. It's when there's clear labeling that's more on one side of the brain than the other, it's fairly trivial to do that, and we can see. But oftentimes when it's more subtle, it uses things like blood vessels in the shape of different structures.
But it is truly using all of the fluorescent signals available to do this.
Yes.
Yeah, so in this case, we get that flipping right every time.
Do this manual interaction. We can see that it's done again. I always look at the ventricles, how well they line up, at how the hippocampus lines up from one section to the next. When I go through all of the sections and I'm satisfied that they're all well aligned, then we move on to the next step. And this is the step in which we can save, we save the stack that's been aligned. And there's different options. The one I'm going to demonstrate is taking those sections and then they're made into a single stack. It's a JPX file. And I'm going to save that.
Yeah, the other thing I'll just call your attention to is that you can change the image resolution that we use all the way up to the original resolution of the image data. It's often good to first save a downsampled version. So in this case, a 25% version, just as sort of a check. It's quick to generate, and then you can take a look and see if the whole structure that was reconstructed looks good, and then you can go and see the full resolution version.
So I'm going to save that. And I'm saving this at a low resolution so that it runs faster, so we can just see, again, for the purposes of demonstration, how that works. I'm going to just do a 25% resolution and then save that.
And so I could talk a little bit while this is going about what this is doing. So we're basically just reconstructing that original brain volume, as it was in the animal, by putting all the sections into the same physical space, and then spacing them out by that intersection spacing. One thing I will also note is that during this procedure, we're resampling the image sections, but only once. And that's really important. We want to make sure that we're preserving as much detail in the resulting image as possible. And so we make sure that when we're doing all of this, that we only do one resampling operation in order to put everything into the same physical space.
And so this is the output. The 3D, viewed in 3D. Again,
for those 7 sections.
This is just a slab of 13 sections of that whole volume.
13 sections, sorry.
Yep. But now I want to show one other important thing that we do. We just close this. What I'm going to show now is that some of the post-processing that we do on these stacks.
Yeah, so I think where you're going to is you're going to show a new feature, a new illumination correction feature that we just recently developed that applies to counterstains or any sort of labeling that has really wide coverage.
Yeah, I'm just searching through the thing. Sorry about that.
Sure. No.
Okay, so this is the full reconstructed brain through all the sections. What I did the demonstration on was just like a slab through this part of the brain, through the thalamus here. So one of the things, if we uncheck the labeling and just look at the Nissl stain. So what we use for our Nissl stain is a fluorescent blue stain called neurotrace blue, and you can see that from section to section, that stain varies. Some sections is brighter, some is less. And also within the section, there's some variability. So over on this side, it's a little bit darker than the other side. This is often happens with these, with stains that are applied to the whole section. And so you can see from section to section, it varies somewhat. And so this is a one that varies quite a bit. It's darker over here, lighter over there. So this new correct illumination function is a modified image function. So we're going to put that on, turn that on here, and turn off the other channels so we don't want to touch. So now we're going to apply this only to this blue channel, which has the Nissl.
So maybe click on and off the preview button a couple of times just to see the impact that it has here.
Yeah.
Yeah. So what's happening is, what this method does is it takes that assumption that the intensity should be even across the section, right? You were pointing out that there was variation in intensity, and what this does is it evaluates how that intensity varies across that section, across the image. And it boosts the intensity in the dim regions, and it diminishes the intensity in bright regions in a smart way to kind of create this more even illumination across the whole section. And that scale parameter that you're working with right now determines how wide of a region is evaluated during that process. And so smaller scales tend to flatten the intensity variations much more than a greater scale. So the larger scale allows for more variation between regions, and a smaller scale basically enforces that all regions will have nearly the same intensity. And the other thing I'll point out here is that in doing this, when this applies, we've actually computed the illumination correction for the whole stack. So you can actually go to a different plane if you'd like to, and see how it applies to any other plane of the image.
Yeah, so now I'm going from one section to the next.
So I think you're probably hitting page 10, and that's actually changing the scale in this case. You might want to use the orthogonal view to go here.
So what Brian's referring to is that there's multiple ways to maneuver through these through the stack, and one is using this ortho view function, and I can go to one section or another. The other is to use this page up and page down, but sometimes it seems like in this case, the page up and page down actually adjusts the scale slider rather than the moving from one section to the other. Is that correct, Brian?
Yeah, that's right. Yeah, and you might want to just turn on preview again, and then you can go through this.
Okay, so this is so if we turn, so I'm going to turn off preview and jump around to for different sections, just so that you can see the variability from section to section. But now if I apply the preview, you can see how that's applied the that illumination correction function to that. And now when I go from the different sections, there's much more. They're much more similar. So this is something that can be applied in the case where we've got like a Nissl stain, which, and what Brian had described, should be evenly applied through all sections and across the entire section. It wouldn't be something that would be used for actually changing the for correcting the illumination of things like the antegrade tracers and all that. Anyway, and so with that, I think that we'll go back to the original. Just to show how. Go ahead.
Chip, just to jump on that point a little bit. So what we're doing is we're really changing the evenness of illumination in the counter stain, which brings out all the architecture for us, so that our eyes can see that in an even way throughout the stack. But we're not really touching any of the tracer data that we've imaged.
Exactly. Yeah, exactly. That's a good point to make. Okay, so that about wraps it up for me. I appreciate you, everyone listening and all. And so now back to you, Nate.
Yep, I'm going to make myself the presenter just because we've gotten a couple questions that I think are really good ones. Let me see if I can figure out the interface. Yeah, so I do want to just point out a few things. This gets to one of the questions. So so everything you're seeing, it's kind of cool. What we do at MBF is a lot of it is born of NIMH grants through the SBIR program, which funds us to make these software tools for the community at large. I want to thank, of course, Dr. Gerfen and Dr. Eastwood and the rest of the team. But this third line gets to something that that is someone asked about, and this link will actually take you to the forums where we've posted notes on the materials for 3D printing these chambers, as well as the files that you could put into most 3D printer programs. So I wanted to address that. And so a couple other questions. Brian, we got a question. What about bright field?
Yep, bright field is handled, no problem. We work with a bunch of labs that do bright field imaging, and we've done reconstructions with those as well.
And that's because you're using a transform that can handle multiple modalities, right? Whether it's confocal or bright field.
Yeah, in this particular instance, the image registration procedure can work with either with either bright field or with fluorescent images, just the same.
Okay, cool. Yeah, that was one question. Yeah, the other one is points to some other webinars that I point you to, and that is we've been asked, is it possible to do this and delimit label cells and delimit specific brain regions where those cells are. And that's we covered that in the last webinar, and I think in the cell detection one we'll go into that a little bit more. All of our webinars are at www.mdfbioscience.com//webinars. So that one is right under the NeuroInfo section of that page. And let's see. We're good on time, so we're almost up, but let's see. We've been asked a few times, like, and I'll just remind folks, we've been asked, can we use, do these have to be confocal images? And these actually aren't confocal images. They're from a wide field epifluorescent Zeiss microscope at 10x. So I'll just reiterate that because we've had a bunch of questions on that.
Yeah, I'll speak to that because yeah, these are definitely all from a epifluorescent wide field Zeiss microscope or Olympus or whatever type of microscope. But we've also used for doing the imaging at slide scanners like the Zeiss Axioscan and those formats. I would emphasize that we use, for most of the things that we've done, using a 10x objective is actually typically sufficient. So even when I showed the those processes and fibers and terminal in the thalamus and all, hopefully you got an idea about how that's very good resolution. So for most things, using a 10x objective is sufficient. The advantage of using for whole brain reconstructions using a 10x objective versus a 20 has to do with the size of the images that are created and how long it takes to do a whole brain. When there are instances where we might have areas that would require imaging at higher resolution, what I typically do is I'll do a whole brain imaging at 10x, and then on those specific regions, we're doing a project now looking at the superior colliculus. I would use a 20x objective reconstructions only through those parts of the brain that would require the higher magnification. And it's not to say we couldn't use confocal images. We could, but again, these have all been done with just epifluorescent wide field fluorescent microscopy.
We got a kind of a procedural question, which I'll answer because it's real easy. Hassan, and it just says, can you send us the link to view previous webinars? And we will. You'll get a follow-up email after the webinar with the survey and various links to the other webinars. And I think we're almost at time.
Am I being viewed? You want me to show the trays?
Oh yeah. Chip, your camera's going, so all you have to do is hold them up.
So here I am. These are the trays. The trays have been done with a 3D printer, and so this is the tray that has the 10 wells in it, 1 mil, and then it fits very snugly into this the tray that has the whole thing. And so this makes doing immunohistochemistry trivial. You can just put 10 mils of the reaction in there, put the slides in, and then shake for overnight or whatever. And then, for doing the rinses, you just take that out, dump things, put it back in. Anyway, so this is this view. And if you have questions, you ask the MBF people.
Yeah, and I think there's some information about what type of plastic to use for the printing, right? Because you found that early on there were some porous ones.
Yeah, it's very important the type of media that's used to do the printing. This is because when some of the media that we used, it absorbed the liquid, and so that's a problem. And so this is, and that's all listed in the information that's provided.
Cool. That's all on the forums here. I've got the link to that here. Just a couple of last questions here. Someone asked, and I want to point this out, is it necessary to do whole brain reconstruction to get your individual sections registered to the Allen Atlas? And no, I just would refer you back to the other webinar where what we actually do with serial sections, because they're somewhat decoupled from each other, even after this rigid registration to each other. When we go to the Allen, we find individual transforms for each serial section into the Allen space. So doing one is kind of where we started with this whole project, actually, and then moved on to 3D volume. So yes, you can definitely do single sections into the Allen.
Like that slab that I showed of just the, the 13 sections through the thalamus, that could have been registered to the Allen space very directly.
And we did a deep dive on our last webinar in that is on our website, which is just www.mbfbioscience.com/webinars. But you'll get a link to those again. The only other thing that came up is what's the minimum number of sections required to create a whole 3D image, and how far apart should they be?
So I should have, we typically section at 50 microns. Well, anywhere between 40 and 80 is typical. And for 50 microns through the whole brain, from the frontal pole into the medulla, is typically about 120 sections for an adult mouse brain.
Cool. Thanks, Chip. And I guess one last really take-home question is, what's the rate of the workflow? Brains per week from front to back, some timings on all of this.
So I can address that as well. So the sectioning in all takes about a half an hour for the brain, per brain. The immunohistochemistry using these trays, and again, if people have questions, I don't mind you contacting me about the trays and all. That's trivial, but it's overnight for doing the immunohistochemistry. Mounting the sections takes anywhere between half an hour to an hour, depending on the expertise of the people doing the mounting. And that's typical, mounting with a paintbrush. People that do histology know how to do that. And so that's, but there's so the manual work of doing the actual histology is half an hour. I always say like half an hour for cutting a brain, an hour for mounting the sections. The imaging for those brains can take, for each slide, can take between 2 to 5 hours, depending on how many different fluorophores there are, how bright the fluorophores are. So the imaging is really the slow part of the process. And so we typically, 2 of us in the lab process. We can process about 2 to 4 brains per week from start to finish.
And getting to the image and the registration.
So I'm sorry, I should have also said that we have 2 microscopes, so we can run 2 microscopes at the same time. Which is, if we have 2 microscopes and and 4 computers, and so we're running a lot of this processing. It takes a lot of it's waiting. We start the program and then it runs. So for this part of the program for the registry or the alignment, the 3D alignment, the manual part of that is when we do the registration. When we do the manual, the manual interactive, the interactive part that Brian described very well. That interactive part is really the slowest part of the process, and that can take between 45 minutes, I would say half an hour to 45 minutes, and then the saving the images can take a while. One other thing I should mention is that we showed saving those images as a JPX stack. You can also save those images as TIFF files, in each section being the individual channels for each section as TIFF files, and those TIFF files then could be used in other programs like Fiji, MATLAB, Photoshop, things like that. So we do a lot of post-processing of sections that have been saved that way.
Cool. Well, we are at the hour. Chip and Brian, thanks again. This is fun as always. And everyone have a great week.